It is my great pleasure to introduce Dr. Heinrich Kovar, PhD, as our speaker for today. Dr. Kovar is a PI at St. Anna's Children's Cancer Research Institute in Vienna, Austria, for which he served as scientific director for 17 years. He is a basic molecular biologist by training who very early in his research career turned to childhood cancer research with a particular focus on bone sarcomas. He discovered CD99 as the immunohistochemical hallmark of Ewing sarcoma in the early 1990s, and subsequently the tools developed in this study were instrumental to the cloning of EWS Fly1 and characterizing this disease on the molecular level. During his scientific career, he made several seminal discoveries regarding the function of EWS Fly1 fusion and its clinical relevance. Dr. Kovar has authored more than 120 publications, and he was awarded an ALSF Crazy 8 grant in 2021. His current research focuses on modeling the origins of Ewing sarcoma, its plasticity, and mechanisms of bone cancer metastasis. So on behalf of ALSF, I wanted to thank Dr. Kovar for his time today, and I'll pass it over to him for his presentation titled The Hen-and-Egg Problem of Ewing Sarcoma Revisited. And as a quick programming note, we have a Q&A session after the presentation, and you may ask Dr. Kovar questions using the chat feature. So with that, I'll pass it to you, Dr. Kovar, and thank you very much for speaking with us today and for your time.
Yes, thank you very much to ALSF and the whole team of ALSF, and welcome from my side to every one of you listening to this seminar today. It's a big pleasure for me to be able to talk about my favorite subject that kept me busy over the last more than 30 years, which is the pathobiology of Ewing sarcoma. And why did it keep me busy? Because as you all know, probably Ewing sarcoma is a very bad disease. It has a high tendency to metastasize, and patients who present with metastases, they still have a very bad prognosis with an overall survival of less than 20%. And those patients that can be rescued from the disease, these are mainly patients with localized disease that don't relapse, they still suffer from lifelong morbidity and side effects of the therapy. So there's really a big need to improve therapy in Ewing sarcoma. And I'm deeply convinced that if we understand the biology of the disease, specifically in the developmental context, we might be able to develop better treatments for the disease. So already for more than 30 years, the main driver of the disease is known. Like most other pediatric cancers, Ewing sarcoma has a acquired genome with very few recurrent mutations, and the only really recurrent mutation is a translocation that you probably all well know, which is the typical translocation between chromosome 22 and chromosome 11 in about 85% of cases, which results in the rearrangement of two genes, the Ewing sarcoma gene encoded on chromosome 22, and then adds transcription factor family gene. In 85% of cases, it is Fly1. In 10%, it is ERK. And in the remaining cases, it's some other family members of these ETS transcription factor family. As a result, a fusion protein is expressed in the host cell, which carries the DNA binding domain of the ETS fusion partner fused to the N-terminus of the EWS protein, an RNA binding protein that has several different functions in RNA metabolism and DNA repair. But in the context of the fusion protein, it provides a potent transcriptional transactivation domain. Now, ever since people have worked on the mechanisms by which the EWS fusion, the EWS Fly1 fusion perturbs the transcriptome of the host cell, and several different mechanisms have been identified. I'm not reviewing all of them, just telling you that EWS1 can both activate as well as repress genes in the genome by several different mechanisms, mostly by binding to chromatin either as a monomer to typical, so canonical ETS transcription factor binding motifs that carry a central GJA core motif, or, and that is very peculiar to EWS fusions, it's very specific to Ewing sarcoma, and not shared with any other transcription factor, whether normal or pathogenic. It can also bind as phase-separated condensates, as multimers, to repeated GGA motifs, to arrays of GGA motifs, GGA microsatellites, which normally are in a closed chromatin conformation, but EWS1 opens them up, and turning them into potent transcriptional enhancers that can activate nearby genes. There are other mechanisms that, as I told you, not going into detail with, just telling you that also it has post-transcriptional activities, it changes alternative splicing, and on top of all of that, it also perturbs DNA double-strand break repair uh, by direct mechanisms binding to DNA double-strand breaks, recruiting PARP1 to the breaks, and also indirectly by sequestering BRCA1 away from DNA double-strand breaks to R loops as a result of transcriptional overflow. So all this is known already for quite some time. And what is also known is that if you introduce EWS1 into different cell types, it has turned out that it is toxic to most cell types. So the question arises, which are the cells that tolerate EWS1 expression? And among those, you would expect to find the origins of Ewing sarcoma that are up to today not well defined at all. Now, over the years, the perception of what might be the developmental origin of Ewing sarcoma has changed with the availability of the of methods of technologies at the time. Like in the 1920s, when James Ewing first described the disease, he described it as an endothelioma of bone. So he supposed endothelial cells to be at the basis of Ewing sarcoma pathogenesis. In the 1980s, based on ultrastructural features as explored by electron microscopy and with the availability of antibodies, more and more antibodies to neural markers, immunohistochemistry, it was found that Ewing sarcoma, despite being a very undifferentiated tumor, still carries some neural features, and therefore a neural crest cell origin has been supposed. Now, more recently, in the 2000s, with the advent of functional genomics, mainly based on RNA interference, and also with the availability of whole genome transcriptomics and also epigenomics, there is accumulating evidence for a mesenchymal stem cell origin of the disease. Now, what I would like to do today with you is to review some studies that we have done in my lab using more recent technologies, stem cell technologies and single cell technologies, and telling you about novel models, and always pointing to the fact that our results depend, and that will be one of the take-home messages, our results really depend on the model that we are choosing. Now, exactly 20 years ago, I wrote a review in which I tried to explain Ewing sarcoma biology, tumor biology, as a result of the oncogene function, of the driver oncogene function within a specific developmental context. And I was discussing the question whether that would be the Hen-and-Egg problem of Ewing sarcoma, whether Ewing sarcoma reflects the phenotype of the tumor stem cell that is blocked in differentiation by the activity of the oncogene, or whether it's the oncogene that imposes an incomplete differentiation program on an otherwise pluripotent precursor cell. Now, ever since the fusion gene has been discovered, people have tried to build a mouse model of Ewing sarcoma by introducing the oncogene into different lineages, into different cell types, both in the mouse embryo as well as in the adult mouse. All of these attempts really failed because the result was either we saw people saw cell death, so as I told you, it is toxic to many cell types, or there was no phenotype, or there was skeletal malformations. And in the adult mouse, what has been seen mostly no phenotype, and if tumors arose, these were sarcomas, but of a different phenotype than Ewing sarcoma. So in my lab, we didn't have any mouse expertise in the lab, and we have no mouse facilities in our institute. So we teamed up with a group of Richard Mohrregel, at that time at the Ludwig Boltzmann Institute for Cancer Research, who was an expert in mouse genetic models. And he had a PhD student, Barbara Sachs, who introduced EWS1 into the mesenchymal lineage early during endochondral bone formation using a PRX1-driven Cre recombinase to activate an EWS1 allele that would be driven by the ROSA26 promoter. And like others have seen, this resulted in perinatal lethality, but the mutant mice, the EWS1 mutant mice, showed severe skeletal malformations, which was the cause of this perinatal lethality. And the skeletal malformations were the result, to cut a long story short, it was really long story, due to the fact that EWS1 was arresting differentiation of the bone formation at an early chondrocytic state. Now, a subsequent PhD student in Richard's lab, Taheri Javaheri, replaced the PRX1 Cre by a tamoxifen-inducible PRX1-driven Cre recombinase. So it would be still expressed, EWS1 would be still expressed in the same lineage, but now in an inducible way. So it was possible by injection of tamoxifen postnatally to induce EWS1 in the adult mouse. And after many years of her PhD thesis, she found out that when she induced EWS1 expression in a narrow time window between day 6 and day 9 after birth, she observed in about 40% of the mice injected the formation of tumors, small blue round cell sarcomas, which the pathologist classified as Ewing sarcoma-like. However, the problem was that these tumors appeared with a latency of 12 to 14 months, which is a nightmare for a PhD student, as you can imagine. So unfortunately, it was impossible to repeat these experiments because the funds were not available anymore. The institute was terminated. The PhD student finished her PhD thesis, and there were other problems. So we could never repeat this experiment. But what it told us, so it remained unpublished. But what it told us was that there's a time component to Ewing sarcoma pathogenesis, which actually doesn't really come as a surprise because we know for a long time that there is a specific age distribution in Ewing sarcoma incidence, with the highest incidence during puberty. It's a typical adolescent tumor occurring with a peak incidence between 15 and 19 years of age. Now, this told us that maybe there is a hormonal component to Ewing sarcoma pathogenesis, to the promotion of Ewing sarcoma genesis. And the hormone that caught our attention the most was insulin-like growth factor 1. Because as you can see here, IGF-1 serum levels change with age with the same age distribution as Ewing sarcoma incidence, as you can see here. In addition, there was already evidence, experimental evidence, already from the 1990s, mainly through the work of Jeff Toretsky and Katja Skotlandi, but also others, that IGF-1 serves an autocrine growth factor function for Ewing sarcoma. Later on, it has been shown that it's actually the fusion oncogene EWS1 that generates an IGF-1 conducive signaling environment. By upregulating IGF-1 expression, by upregulating papalysin A, which is a protease that cleaves IGF binding proteins, which are negative regulators of IGF signaling. And there was also evidence from several in vitro studies that if you block IGF-1 signaling in Ewing sarcoma cell lines in vitro, this would kill the cells. However, in vivo, in clinical trials, only 10 to 14% of patients responded to anti-IGF-1 receptor therapies. So a subsequent PhD student now in my lab, after the Ludwig Boltzmann Institute had been terminated and Richard was moving on, Rahil Norisadih, she performed the following experiment. She went to the freezer and she took out cells that were isolated by Taheri Javaheri from the limbs of the mutant mice. which we called mesenchymal stem cell-like cells because they were from the limbs of those mutant mice. She also isolated mesenchymal stem cells from the limbs of wild-type mice. So the wild-type don't express EWS1, the mutant ones express EWS1. All experiments were performed with mesenchymal stem cell-like cells from 2 different mice. And she put these cells into culture, and then after expanding them into soft agar. Now, soft agar assays, you might know, are a surrogate for malignant transformation, an in vitro surrogate for malignant transformation, which tests the clonogenic potential in a surface-independent way of cells that might be tumorigenic. And she did so in the absence of any hormone treatment or by adding 400 nanogram per ml, which are the serum concentrations during puberty in the humans of IGF-1, or 100 nanogram per ml insulin, or a combination of IGF-1 and insulin during the soft agar incubation period. And to our big satisfaction and in a way surprise, what she saw was that in the hormone-treated condition, there was a strong increase in colony numbers. So the untreated ones hardly formed any colonies, but the hormone-treated ones formed colonies. So she now took those what we call hormone-activated EWS1 expressing mesenchymal stem cell-like cells from the colonies. She expanded them again, but now in the absence of any hormone treatment, she put them into soft agar again, and again kept them in soft agar, incubated them in soft agar for 3 to 4 weeks, still in the absence of any further hormone treatment. And to our big surprise, the cells kept their transformed phenotypes. They still formed tumors with high efficiency colonies in soft agar with very high efficiency, telling us that the transient hormone exposure stably reprogrammed those cells to a transformed phenotype. Importantly, when she now took these cells and injected them into mice, they formed tumors with every injection very efficiently, much more than the hormone untreated EWS expressing cells did. And those tumors were of a small blue round cell phenotype, as you can see here. Now, Rahil generated a lot of RNA sequencing data and also chromatin accessibility data, ATAC-seq data, to further characterize each individual state in this transformation process. And Florian Halbritter, the computational PI at our institute, had a closer look to the chromatin occupancy data, the ATAC-seq data. And what he did was he clustered the chromatin regions that are differentially open in the different conditions according to the specific behaviors. And he found he could define 5 clusters. And cluster 1, as you can see, are those chromatin regions that are open in the wild type situation, even if treated with IGF-1, but closed in the EWS1 mutant condition, no matter whether treated or not treated with IGF-1. Similarly, cluster 4 shows the genes on the chromatin regions that are closed in the wild type, but open in the EWS1 expressing condition, no matter whether treated with IGF-1 or not. And there is another cluster which is kind of similar. It's closed in the wild type, it's open in the EWS1 mutant condition, but it closes down with a hormone treatment again. But most interesting to us were these 2 clusters, M3 and M2. M3 showing genes that are open in the wild type, also remaining open with EWS1 expression, but only closing down in the context of EWS1 expression and hormone treatment. And vice versa, M2, which is closed in the wild type, remains closed in the EWS1 mutant condition, but only open up upon further hormone treatment. And this small cluster M2 was most interesting, as it turns out that these regions are next to well-known Ewing sarcoma marker genes that are also dependency genes for Ewing sarcoma, like BCL11B, protein kinase C beta, R-spondin, SOX2. And also we saw an upregulation of YAP1. YAP1 is a transcriptional co-activator downstream of hippo signaling, which regulates the activity of teeth transcription factors. And interestingly, when Florian did motif enrichment analysis in the differentially open regions of the different clusters, he found that cluster 4, this one over here that is open in the presence of EWS FLAIR1, is heavily enriched in teeth, and this is the most enriched motif in teeth binding motifs, suggesting that maybe it requires YAP1 to activate those genes in this cluster that are opened up by EWS FLAIR1. And this cluster contains a number of genes that are well-known genes in self-renewal and stemness genes like cyclin D1, KLF5, MET, and so forth. So Rahil tested several of those genes for the dependency on YAP1 expression. So she did knockout of YAP1 using sgRNA against YAP1, so CRISPR-based knockout. And as you can see here, for both mice, these genes were all dependent on YAP1 expression, as was colony formation, because in the YAP1 knockout, colony formation in the hormone reprogrammed cells was lost. Also true for both mice, as shown here. But do these cells still depend on EWS1? And the answer is yes, they do. Colony formation was also lost upon knockdown of EWS1. So that means the hormone reprogrammed EWS1 expressing mesenchymal stem cell-like cells depend on both YAP1 expression and EWS1 expression. Now, this brought us to put forward the following model, the following hypotheses. We think that early during Ewing sarcoma genesis, maybe when the translocation occurs, maybe embryonally already, EWS1 gets expressed. It does what it does to the transcriptome, so it screws up the transcriptome by the various mechanisms that I told you, and among them, it upregulates the autoregulatory IGF-1 feedback loop, which keeps cells going that they survive, because these cells, I forgot to tell you, these are immortal, but they are not transformed. They cannot form tumors. Later on during the development, maybe during puberty, when IGF-1 and insulin levels increase, they are able to activate not only the IGF-1 receptor, but also possibly the insulin receptor. The insulin receptor is a low affinity receptor for IGF-1, which allows the activation of several genes, including the YAP1 gene by a mechanism that is not yet fully clear, which allows then to co-activate genes that are bound by that are opened up by EWS1, recruiting the T transcription factors, and will then get activated by recruitment of YAP1, which then promotes self-renewal, stemness, and malignancy. But a model is just a model. And so we wanted to know whether we can get any evidence that mesenchymal stem cells from the bone marrow of patients could be the basis of Ewing sarcoma genesis. And for that, in the context of the Crazy 8 initiative funded project, and we are very grateful to the ALSF Foundation for choosing us with this project for the Crazy 8 initiative, Utkarsh Kapur, a very talented postdoc in my lab, and Christoph Havemeister, a computational biologist in Florian Halbrichter's lab, set out to establish a single cell atlas of normal and EWS1 perturbed mesenchymal stem cell differentiation to figure out if there is any stage in human healthy juvenile bone marrow mesenchymal stem cell differentiation that would allow mapping of Ewing sarcoma primary transcriptomes to it. So that was the whole purpose of the exercise. The experimental outline is shown here. We collected healthy mesenchymal stem cells from the bone marrow of healthy donors, all below the age of 18 years during a surgical procedure that is called temporary epiphysiothesis of the knee joint. Both female and male donors, altogether 19 donors. We generated pools from those donors, which were then transduced with an inducible EWS1 expression vector that also carries a GFP cassette that allows selection of those successfully transduced cells, which were then put into micropatterned culture dishes to allow for cultivation and differentiation of the mesenchymal stem cells as spheroid cultures in spheroid conditions, also under low oxygen conditions, because we assume that these are more physiological conditions to test. And then Utkarsh induced EWS1 expression on a daily basis during the differentiation in a serum-free differentiation approach into each of 4 lineages. And then samples were collected every day for the control conditions and the EWS1 induced conditions, and EWS1 was expressed for different periods of time. So it's a big experiment, and the samples were then subjected to 10x single-cell transcriptome sequencing. And here you see the map that Christoph obtained. You can clearly see that the 4 lineages can be distinguished from the parental mesenchymal stem cells. And here you can see what happens upon EWS1 expression. There is a clear perturbation of the transcriptomes of these differentiating cell types. Now, all this, what I've shown you so far, depended on the pre-definition of a candidate cell type of origin, which was mesenchymal stem cells from the bone marrow of a vertebrate organism. But we wanted to know whether we have any method, any means to identify the Ewing Sarcoma progenitor cell in a completely lineage ignorant, in an unbiased fashion. And for that, again, as part of the Crazy 8 project, Sarah Griesenberger, a very talented PhD student in Martin Distel's lab when Martin was still at our institute, he has recently moved to Huntsman Institute, set out to come up with the following experiment. And this was based on the hypothesis of Ewing sarcoma, similar to adult cancers where it has already been demonstrated, keeps an epigenetic memory of its lineage origin, of its cell type of origin. And the epigenetic memory, we expect it to reside in enhancers that are open in Ewing sarcoma independent of the expression of the oncogene. So, and which we would expect to be also open in the cell type of origin, or that are repressed by EWS1. So they are closed in and not active in the tumors, but might have been active in the cell type of origin, just closed by the activity of the fusion oncogene. And so she mined a number of data sets that we had produced over time, methylation data sets, ChIP-seq data sets for histone marks, ATAC-seq data, CAGE-seq, which is a method to identify enhancer RNAs, to come up in the end with 46 candidate enhancers. We did not look at the genes that they would drive. We were just looking at the enhancers themselves. And she took those enhancers and cloned them individually in front of a fluorescent protein expression cassette, which was then introduced into zebrafish larvae at the 2-cell stage to follow their activity over time during the zebrafish development, with the idea of looking for cell types on which individual enhancers would enhance activities would converge, and then dissociate those larvae and FACS sort those fluorescent cells and subject them to single cell sequencing to characterize the single cell transcriptomes and get a clue about their lineage and their identity and their developmental stage. She would also take some of those enhancers that are representative for that potential epigenetic memory enhancers to drive EWS1 and test for any phenotype that they could see in the adult fish. I can tell you, we have done the single cell sequencing. It turned out to be a lot more complicated than we thought, mainly due to difficulties in aligning the individual transcriptomes to specific cell types in reference atlases, and this analysis is still ongoing. But while this analysis is still ongoing, we wanted to validate these findings again in the human system, and therefore UTCARS has started to differentiate using a protocol that has been published by Xi et al. Human-induced pluripotent stem cells in presomatic mesoderm and nascent somites, and from there to sclerotome and dermomyotome, and from there in the different lineage, in the 4 different lineages, in the absence and again induced presence of EWS1. And so this experiment has already been done, but again, actually the samples are currently running on the sequencer. So I cannot tell you the outcome of this experiment. But the question is, can these enhancers also drive tumorigenesis if used to EWS1? And the answer is yes. So far, tumors formed from enhancers with expression in somites. And here you see an example for a tumor that formed over here, which also was found by Sarah to express CD99, which, as you have heard in the introduction, is a hallmark of human Ewing sarcoma. Now, in addition, Adam Varady, another very talented PhD student in Martin's lab, had developed an optogenetic method to allow for gene regulation in a spatially confined, spatially very restricted manner. And when he activated EWS1 in different areas of the body of the larvae, he could find that not only tumors formed, but if he irradiated, for example, the caudal fin to activate EWS1 or the dorsal fin fold, then accessory additional ectopic fin formation was observed, as you can see here and here. And at the basis, you can see the EWS1 expression. While when he irradiated with light and activated EWS1 in other areas of the body, in the trunk, for example, and the head, he didn't see this additional fin formation, but rather tumor formation, which told us that there is a position effect for EWS1 in the body of a vertebrate organism like the zebrafish. But again, a model is just a model. And so we turned back to the human system, asking the question, what happened? Yeah, because so far we have not considered that that there might be a dose effect of EWS1. So we are always expressing a transgenic or a knock-in EWS1 allele, but never consider that there might be a dose effect. And why is the dose effect important? Because it has already been shown very early on by Franck Tirode when he was still working in Olivier Delatre's lab, and that was actually the seminal publication that was taken as an indication for mesenchymal stem cells being at the basis of Ewing sarcomagenesis, that if one knocks down EWS1 expression in Ewing sarcoma cell lines, that transcriptomes move close to mesenchymal stem cells. Now, several labs subsequently, and the most frequently cited paper in this context is the one by Francetti, also from Olivier Delatre's lab, showed that actually if one modulates EWS1 expression, it leads to an epidermal mesenchymal transition-like phenotype. So it makes the tumor cells much more mesenchymal and shuts down proliferation. That was seen by several labs, including our own. And actually, the lab by Kim Stegmaier showed that in order for Ewing sarcoma cells to proliferate, there needs to be an optimal EWS1 concentration, which she called the Goldilocks principle in Ewing sarcoma. So this brings us back to the Hen-and-Egg problem of Ewing sarcoma, because the question is this mesenchymal phenotype a phenotype of the cell of origin, or is it still connected to the activity of the EWS1 fusion oncogene? And so
Vivian Suresh, a very talented PhD student in my lab, and Valerie Fock, a postdoc in my lab, set out to do the following experiment. They knocked into the endogenous EWS1 gene locus a fluorescent tag as well as a degron tag, which would be activated by addition of a DTEC ligand, a VHL ligand, which leads to rapid degradation of the fusion oncoprotein. And by adding different concentrations of the DTEC ligand, they could actually establish a gradient. They could really titrate EWS1 concentrations. They could establish a gradient that could be maintained for weeks at the same level of EWS1 expression. You see this here by flow cytometry. You can see this here on the Western blot for the different DTEC concentrations quantified here. And here this table tells you that they could establish levels of 86%, 65%, 43, 16, and 7% of the original EWS1 levels in the cell lines. So what happens? Phenotypically, we see that the Goldilocks principle also applies to typical EMT phenotypes like increased migration that we can see has an optimum here in two different clones of that cell line, around 50, a reduction of EWS1 by around 50%. As you can see here, same is true for invasion here using an organ on a chip assay. You can see here when Vivian reduced the expression of EWS1 by 35% or by 47%, you see the highest level of invasion, which then goes down again with higher concentrations of the DTEC ligand, while at the same time, clonogenic growth in soft agar was severely affected already with minor modulation of EWS1 expression and was lost already with an introduction of 35 to 50, 60% of EWS1. So the question then arose, so that is nice, but what if EWS1 would actually fluctuate? And there was some evidence, preliminary evidence from the lab of I know et al. And also from Kim Stegmeyer's lab that there might be fluctuations of EWS1. So can we model the fluctuations? And for that, Vivian and Valerie took the cells that had been modulated EWS1 expression for 1, 2, 3, and 4 weeks. You can see that this results in severe changes in the transcriptome as expected. That is also shown here in this PC plot with you see there's a concentration dependent effect and also a time dependent effect. And now they washed out the ligand, which leads to very rapid recovery of EWS1 levels to 100% within 16 hours, as you can see here. And surprisingly, the transcriptomes did not go back to the baseline that would be here, but rather a large number of, or not large, but a number of genes remained mainly upregulated. Some of them also remained downregulated persistently, even though EWS has already recovered for 1 week. Now, what are the consequences for the phenotype of the cells? Again, we can see that the migratory phenotype is not fully lost. In fact, it is maintained at a lower level, but it is still maintained. Same is true for the invasive phenotype. The only difference here is that during the phase of EWS1 modulation, the cells invaded as single cells, while after recovery, they invaded collectively. You can clearly see this and very nicely see this in this organ on a chip assay. However, the colony forming ability was regained upon rescue of EWS1 expression, which means that the cells end up in what is known from adult cancers as a hybrid EMT phenotype, a hybrid epithelial mesenchymal transition phenotype, which is well known to promote metastasis. And in fact, what we see is both in the fish model, we see increased extravasation, and in the mouse model, we see increased lung metastasis upon tail vein injection, which is seen here again with a small round cell phenotype. So in fact, that means that a transient modulation of EWS1, even a moderate modulation of EWS1 expression, results in a more malignant phenotype, highly metastatic phenotype, which might have clinical relevance. And interestingly, of something like 100 genes, a little more than 100 genes that show this permanent change, about 20% showed significant association with adverse prognosis in the cohort of 196 patients collected by the Thomas Grunewald lab. So finally, I just wanted to mention that we're also interested in what are the upstream mechanisms that may result in fluctuations of EWS1 and modulation of EWS1 expression. To analyze this, Vivian performed a high throughput compound screen and also a high throughput CRISPR screen. We ended up with a number of candidate pathways. This is still under analysis, but you can clearly see that there are physiological pathways that affect EWS1 levels. And yes, and this is still under investigation. So to sum up from what I've told you today in a nutshell is that we think that Ewing sarcoma phenotype is influenced not only by the cell type of origin, but also by the body localization and the developmental stage of the cell of origin, the organismal developmental stage at the tumor initiation, as well as the developmental stage and the endocrine milieu during tumor promotion. So that was the IGF-1 story. The high throughput screens tell us that there are physical and other signaling microenvironmental cues that affect Ewing sarcoma, EWS1 expression and Ewing sarcoma phenotype. And importantly, the temporal dynamics and the amplitude of EWS1 fluctuations have an effect on Ewing sarcoma phenotype as well. In the end, what I wanted to show you is that all these results really depend on the model that you use. And please keep in mind, a model is just a model and it needs to be validated in the human as well. So with this, I would like to close and take questions and thank all the members of my lab, a wonderful team. And I'm very grateful to work with this team, the members of the Martin Distel lab, and we are very sorry that we lost him to the Huntsman Institute, but I'm sure that he will be very successful there. The Halbritter lab and the bioinformatics core facility, and collaborators at universities in Vienna and other institutions. I cannot name all of them. And thank you for your attention. I'm happy to take questions.
Great. Thank you, Dr. Kovar. As we mentioned before, I encourage individuals who have questions to please post them in the Q&A box, and we'll read them to Dr. Kovar and hear the responses. So the first question involves IGF-1 production. You mentioned age as where you see enhanced IGF-1 production. Are there any other factors that influence IGF-1 production? And could that be a prognostic indicator of progression of Ewing sarcoma outside of just age?
So, yes, so what is known already is that physical stress in the bone, for example, increases IGF-1 expression in the bone environment. Which might come together with a frequent observation that patients are frequently diagnosed after an insult at sports and so forth, and it also comes together with a physical activity of adolescents. So that is something that is known and might have an influence there.
Okay, thank you. Our second question is it known how the specific hormone treatment affects the different cell types, and what would that effect have on the tumorigenicity of the cell? So how might hormone treatment impact the different cell types, and what impact might that have on tumorigenicity of the cells?
In the mesenchymal stem cell situation, not in the mouse model, in the mesenchymal stem cell, human mesenchymal stem cell situation, that's something that we are currently testing. So we are testing the different lineages for the response to IGF-1 treatment. We can see for the undifferentiated mesenchymal stem cells that their clonogenicity increases. For the different lineages, this is still ongoing. So I cannot answer this for sure at this point.
Okay, thank you. And another question is, EWS Fly1 acts both at developmental enhancers with ETS motifs and GGAA repeat elements. The positions of the latter are less well conserved in model organisms. Do you see differences in the epigenetics or phenotype of your human versus non-human EWS models that might be attributable to those different mechanisms?
Yeah, that is a really very relevant and good question. Thank you for that. So what we can say is, yes, definitely there are differences in the GGA microsatellite landscape. Not only in number, actually mice have more GGA microsatellites than humans, for example. Fish have less. The length of the GGA microsatellites are also longer in mice than in human. We see the GGA microsatellites satellites at different locations with respect to the genes, to the next genes in the neighborhood. We have not systematically looked at differences in EWS Fly1 occupied sites with respect to GGA motifs in the mouse and in the human and in the zebrafish yet.
Okay, thank you. So another question revolves around the Goldilocks paradigm that you see with EWS Fly1 expression. So within patients, is there any stratification even within that Goldilocks range, or do you see very consistent expression of EWS Fly1 within patients?
So what is known from patients, so let's see, the bulk tumor cell population in the patient has a very consistent EWS1 expression. So there is not much variation. However, there is evidence that a small proportion of cells in the range of few percent might have different EWS1 levels. Whether this is transient or not needs to be demonstrated. Whether there are conditions where more cells show a different concentration of the fusion oncogene is not known specifically. The hypothesis would be that on its way to settle down as metastasis, the cells might have lower EWS1 expression, but it's difficult to find these cells. One would really have to look to cells in the circulation, but to my knowledge, these studies have not yet been performed.
Okay, thank you. So are there any other questions? Please continue to post them in the chat. So right now we have kind of one more in the queue. And going back to the question of the positional effects with Fly1 expression, in terms of, could you further elaborate on the role of what you hypothesize might be attributable to treatment with the adipocytes and how that might be helpful in informing treatments to have expression of EWS among those type of cells?
So not quite sure whether I got the question right? So the whether the information that we get that adipogenic lineage might be the cell type of origin, whether this would have any influence on the treatment.
Yes.
Is that the question? Yes. So I think we cannot say this at this point. This is definitely what we would like to explore. Yes, so the answer is I cannot really answer this at this point.
Okay.
Yeah. With regards to the position effect within the organism, as I told you during the talk, the single cell sequencing results actually that are still under analysis highlighted, despite showing this clear limitation to specific cell types like the muscle and the somites, for example, when we flow sort the cells based on their fluorescence, we actually see a lot more cell types in there. And these are developmental cell types, and whether in different body positions, the composition of this mixture of cells might be different, and we cannot say at this point, and which cells specifically are the ones then in which the enhancers are really active. We can only say if we have really thoroughly analyzed the single cell data from different positions in the body.